Hello everyone, welcome back to FST3711 Food and Industrial Microbiology. Today we are going to continue with the taxonomic groupings of microorganisms that are important in foods, and we are going to talk about molds. Some learning outcomes after this section. You should be able to group different types of microorganisms into the different groups of molds, yeast, and bacteria, and be able to differentiate the groups according to morphology and characteristics. You should also be able to relate the growth conditions to food these microorganisms can spoil, and describe the types of food that are susceptible to the different types of spoilage by the different taxonomic groups. And we are going to also talk about the enumeration of molds and bacteria towards the end. So, what are molds? They are eukaryote, multicellular, and they produce spores. They are also known as heterotrophic organisms, as they require organic carbon as an energy source. Most of them produce extracellular enzymes and absorb their nutrition through hyphae and mycelia. Some of them may produce toxins and toxic compounds that could be carcinogenic. Shown here are various types of molds, or also known as fungi. One important characteristic of fungi is that they lack chlorophyll, and this characteristic profoundly affects the lifestyle. They do not depend on light for survival, and this means they can occupy dark habitats, which literally can be anywhere, everywhere. They can grow in any direction, three-dimensional growth, and can invade the interior of the substrate with absorptive filaments, which is hyphae, which you will see a bit later on in detail. Now, a mold is the common term or layman term that we use, can be referring to three different types of fungi. A mushroom produces fruiting body, and the fruiting body usually contains spores, and this is shown in the bottom picture. A mold or a fungus is a filamentous fungus generally that do not produce a microscopic fruiting body, and normally seen as powdery growth on food and foodstuff, like shown in the top picture. A yeast is also fungi, and this is normally unicellular in growth, and they are non-filamentous. Let's look at the morphology in the vegetative structure. A lot of them are multicellular when we are talking about fungi. They are filamentous or can be dimorphic, meaning there are two forms of growth in the life cycle. They can change from yeast-like to mold-like or filamentous-like. Now hypha is singular, hyphae is plural, and this is the basic cellular unit in filamentous fungi. Collectively, they are called mycelium. Mycelia is plural. There are basically two types of mycelium, the aerial mycelium or the penetrative, generally horizontal type of mycelia. Septa are the cross walls that divide the hyphae into uninucleated or multinucleated cells. And there are also fungi that can be non-septated or aseptate. Now let's look at a hyphae. This looks like a growth of a young fungal colony arising from a single spore, the black dot in the middle. How many hyphal tips do you think there are in this illustration? It's just a hypothetical question. You don't need to answer this. The hyphae is a very strong structure for filamentous fungi. On top of that, they are also waterproof and made up of chitin. The way they grow are always in a branched pattern, and at its growing tips they secrete digestive enzymes that will break down complex molecules into simple molecules that the hyphae will absorb as nutrition. And inside the hyphae there are hydrostatic pressures, and these make it ideally suited for penetrating, exploring, and exploiting solid substrates in a manner that other organisms like bacteria may not be able to match. Successive hyphae branching and hyphae fusion can occur in the interior of the colony and become dense in the middle, and this results in a complex synocytic multicellular network that nutrients are acquired at the colony periphery and can be effectively distributed to the distal regions and used to support reproductive development. Hyphae come in two complementary mating types, and we normally label them as plus strand or minus strand, as we can't easily define or label them as male or female type. This is the classification of a white button mushroom in the family of Agaricaceae. The genus is Agaricus and the species is Bisporus. This is the common white button mushroom that you can buy off the shelves of supermarkets. A few phyla of fungi will be discussed here. For example, the Ascomycota, Zygomycota, Basidiomycota, and the Deuteromycota. Actually, there's no Deuteromycota phylum anymore in the new classification of fungi. Now, mainly of importance to food spoilage are the Ascomycota and the Zygomycota phyla. That includes their imperfect or asexual or anamorph stages. Basidiomycota or Basidiomycetes are actually species that produce fruiting bodies that are generally collected as food. We look at the reproductive structures of fungi that includes asexual and sexual structures. These are markedly more complex compared to the simpler bacteria and archaea. We have teleomorphic life cycle where there's a sexual reproduction stage, and there's also the anamorphic cycle where the imperfect or mitotic asexual stage, and many fungi, to make it more complicating, many fungi can have both states, especially Ascomycota, but most have either one or the other cycles. Two main reproductive units are spores and fruiting body. Spores are carried by fungi, all fungi, as the minute propagative unit functioning as a seed, but they differ from seeds in that spores do not contain a preformed embryo. In fungi that produces fruiting body, this particular structure contains or bears spores and is called a sporocarp. And these are mainly in the mushrooms that you see at the bottom pictures. Now this slide shows classification of fungi that is done based on their reproductive structures. Fungi imperfecti are fungi that are not capable of forming sexual spores, and the other four types are able to form sexual spores, resulting from the fusion of nucleus from two opposite mating strains of the same species of fungus. If the hyphae is septated, they are probably either ascomycetes or basidiomycetes. If the hyphae are non-septated, they are most likely zygomycetes or oomycetes. To look into the detail, these are what the sexual spores of the three main types of fungi look like. For zygospore, they are like a black ball, sort of squeezed in between two bulging hyphae. Picture on the top shows zygospore, a growing zygospore, and an ascospore is a spore that is enclosed inside an ascus. Generally, there are 4 or 8 ascospores in an ascus, shown in the second picture below zygospore. And the third type is the basidiospore that are produced on top of a basidium, a bat-shaped basidium. And generally there are 4 basidiospores for every basidium. We look at the overview of the different terms that are used for their reproductive structure. In zygomycetes, The sexual reproduction produces zygospores, whereas the asexual reproduction will produce sporangiospores. As for ascomycetes, which is commonly known as the sac fungi, sexual reproduction produces ascospores, and the asexual reproduction produces conidiospores. For basidiomycetes, club fungi, sexual reproduction produces basidiospores, and the deuteromycetes, which are fungi imperfecti, produces only asexual reproduction. Produces conidiospores in asexual reproduction. In the kingdom Chromista, which is related to fungi, oomycetes are water moulds that also have sexual and asexual reproduction. During sexual reproduction, they will produce oospores, and during asexual reproduction, they will produce zoospores. However, these fungi are not very important in food, so we will not go into too detail about this group. In this phylum of Ascomycota, we have yeast, cup fungi, truffles, powdery mildew, and morels. We will talk about yeast separately in the next part of the lecture. Here we look at the fruiting body type of ascomycetes. The sexual spores are ascospores produced in sac-like structure called ascus, generally containing 8 spores per ascus. They are anamorphs, and this includes the fungi imperfecti Deuteromycota. Used to be in Deuteromycota phylum. The asexual spores are usually conidiospores, and sexual spores are called ascospores. The two most important examples of anamorphs are the Penicillium species and Aspergillus species. In the phylum Zygomycota, these are the common bread mold that you see, and they have synocytic non-septated hyphae as a main characteristic. They are also easily recognizable by their formation of sporangium and sporangiophore, carrying dark black tiny spores. They are important food spoilage molds, for example Rhizopus and Mucor species. They usually produce asexual sporangiospores as shown in the right picture. Some produce sexual zygospores where they are thick wall and resist drying, but not well adapted for disposal. But its resistance to drying is an important characteristic when we talk about food spoilage. This phylum Basidiomycota contains the club fungi, so called because the basidium looks like a club-shaped bed. Basidiomycetes include mushrooms, toadstools, puffballs, bucket fungi, shelf fungi, stinkhorns, rusts, and smuts. Some are used as food, for example mushrooms, but others can cause crop damage, such as rusts and smuts. They seldom reproduce asexually, and the basidiocarp is made up of a stalk called the stipe and a flattened cap. The stipe may have a skirt-like ring below the cap called the annulus, and there are gills found on the underside of the cap. And these are lined with basidia, and the basidium refers to the sexual reproductive structure that makes the basidiospores. I'll show you some pictures later. Basidiospores are released from the gills and germinate to form new hyphae and mycelia, And these vegetative structures are found below ground and include rhizoids that anchor and help absorb the nutrients from the soil or the tree trunk and roots, and hyphae and mycelia as well. Next, just to touch a bit on the fungi imperfecti, which has no known sexual stage in their life cycle. Most of these species reproduce by conidia, and they are classed by color or structure of the conidia. Since most of the conidia structures look like ascomycete type, they are believed to be derived from the ascos who lost the ability to reproduce sexually. In some cases, however, their forms look like they may have been derived from a basidiomycete. And commercially important species include those from the Penicillium genus, such as Penicillium notatum, which produces penicillin, Penicillium chrysophovum, that produces antifungal that are used in ringworm and athlete's foot treatment, and the Penicillium roqueforti and camembertii for their cheese production. This is just to show the different types of asexual spores from fertile hyphae of different types of fungi. We have the common conidiospores produced by Penicillium species, and these are not enclosed in a sac. Arthrospores formed by Geotrichum species, formed by fragmentation of septated hyphae into single cells. Blastospores are buds from parent cells found in some yeast, such as Candida albicans and Cryptococcus species. Chlamydospores are thick-walled spores formed by rounding and enlargement within a hyphal segment, formed by Candida albicans. And sporangiospores are also common from Rhizopus species. These are formed within a sac called sporangium, and these sporangia are formed at the end of aerial hyphae called sporangiospores. Here are some pictures to illustrate what we mentioned earlier in a few slides before. The picture of far left is a picture of conidia formed in a bunch on top of a conidiophore, like for the Aspergillus species. And here we have arthrospores, top red color tinted picture formed by septated fungi. At the bottom, C. Blastoconidia formed by yeast, certain types of yeast, and D. Chlamydospores on the ends of hyphae-like structures. For sporangiospores, these are numerous tiny spores that are formed inside a sporangium on top of a sporangiophore, which can be branched or unbranched, and they all differ in size, shape, color, and location according to the different species of zygomycetes. In sporangiospores, such as mucor species, you can see this typical columella underneath the sporangium that will collapse when the spores are released And shown here at the bottom is a close-up picture of the sporangium formed on an orange. The rest are pictures, micrographs, seen under microscopes. When you grow these molds in culture media, you will see loose or compact type of growth as shown here in the picture. And there are, of course, various sizes and color of colony based on the different species of fungi. How would you count fungi, knowing that they have millions of spores? And this is quite difficult as the mycelia does not fragment into colony-forming units. You don't normally count fungi as easily as CFU for bacteria. Hence, we underestimate most of the time. Spore numbers can be very high as a significant contributor to growth, but there might be few colonies growing on the plate. And no one medium is useful for isolation and enumeration of all molds from all foods. Hence, we look at methods that may not just rely on the growth of fungi on culture media. Here we look at a slide wet mount, which is an early method derived in 1930s based on bacterial detection. We grow them on proteinodextrose agar acidified to pH 3.5 with tartaric acid, and later we replace the medium with antibiotics, chemicals or dyes to get pure cultures of fungi. For internal mold examination, we do direct plating generally, where this is mainly from plants. You must do a surface decontamination of intact seeds, nuts or grains by rinsing with bleach or alcohol, and then sometimes rinse off with sterile water. After that, you plate the grains on appropriate agar culture at 25 degrees Celsius for about 5 days to see colonies of fungi, and you can also count them. However, there are limitations to all these methods, as sometimes there are inability for us to decontaminate heavily contaminated surfaces. Sometimes the alcohol used can be absorbed by grain and the molds get killed, or the bleach is also killing off the fungi that we intend to isolate. Organic matter on the substrate can also degrade chlorine, and seeds and grains can germinate on the agar, preventing the molds from growing. For total mold count, generally, we still use the culture media to help grow and maintain the molds and yeast, and we generally use a sample size of between 5 to 400 grams depending on how homogenized the sample is, and we blend it in a stomacher and dilute if necessary. After that, we spread plate on the appropriate agar media and we incubate, and colonies that grow between 20 to 200 will be counted and recorded as CFU per gram or per mil of the sample. This is for fungi from food samples. There's a scheme of method shown here. Generally, microscopy is done if there's obvious growth of fungi in your food, or else we will do direct plating or dilution plating as shown in this scheme.